It's a great pleasure to welcome you to the first public lecture of the British Pharmacological Society, and it's a particular pleasure to welcome our first speaker, Leslie Iverson, to give his talk on bringing cannabis back into the medicine cabinet.
It's a pleasure to be invited to give the first public lecture to BPS. I'd like to make it clear I'm speaking here as a private citizen, a retired professor of pharmacology, not in my capacity as the chairman of the Advisory Council on Misuse of Drugs, and I will refuse to answer any questions about recreational uses of cannabis or its classification, which the Advisory Council have considered on 3 different occasions in the last 10 years, and I hope I never hear about it again. What I'm going to talk about instead is the medical uses of cannabis and the revival of interest that we see around the world in this aspect of the plant. And I'll tell you what's happening in this country and some of the snapshots from around the world. And my lecture, being a public lecture, pharmacologists may be disappointed because there won't be very much science in the lecture, very few chemical structures, and more emphasizing the social history and the medical history of the subject. Subject is the product of the cannabis plant, a herbal material in America known as marijuana, and the actual drug is secreted by bracts around the flowering heads of the plant, particularly concentrated in the female flowering heads. The male flowering heads are a nuisance because they get in the way and don't produce much drug, and this male plants also allow the female plants to make seeds, and they get in the way of cannabis smokers. Most cannabis is now grown as a female plant form. And I'll show you later a growing facility in the south of England, the only legal one we have in this country, where all the plants are female and they're all cloned from one single female plant. For gardeners in the audience, this means they're all reproduced by cuttings. Cloning sounds a much better word, but that's what it is. And the active ingredient, psychoactive ingredient in the cannabis resin is this lovely creature, delta-9 THC, first described in the early 1970s by Raphael Makhoum in Israel. The pure THC is a thick, greasy, viscous treacle, very hard to handle and very difficult to administer because it's totally insoluble in water, and there are no polar groups, so you can't make salts of this chemical. And we'll come to how you can administer it in a moment. But that's the active ingredient. There are about 50 other so-called herbal cannabinoids which have similar ring structures, but nearly all of them are inactive in terms of their intoxicant properties and inactive on the receptor, which I'll tell you about in a moment. The history of medical cannabis goes back a very long way, thousands of years in China, and particularly in India. And this gentleman, William O'Shaughnessy, was a Victorian brilliant genius who brought cannabis back from India. He was a remarkable man. He was trained as a doctor, but already at the age of 22, during his training, had the idea for treating cholera with fluid electrolyte balanced replacement, which is still the treatment for cholera today, and all those diseases characterized by severe diarrhea, dysentery, etc. And this is a brilliant step forward in medical treatment. It's very cheap and still very widely used. But he went on, joined the East India Company as it was in those days. So he was a physician, surgeon, professor of chemistry, and also a research scientist. And the research scientist bit was very important for the cannabis story because he got interested in the folk use of cannabis in India and started systematically studying it in sort of medical trials using some animal research, basic research, and convinced himself that there was something in the story, that cannabis actually did have some medical benefits. And in 1841, when he returned to India, he brought some cannabis with him and generally popularized the whole idea of cannabis as a medicine, which took off very rapidly in the 19th century when very few other medicines were around. And I'll show you some of the preparations that were made and widely marketed. But Shaughnessy wasn't finished. He went back to India and among other achievements, installed 3,500 miles of telegraph network throughout the entire continent, which was extremely useful to the British in putting down some of the native mutinies at the time, and just another remarkable achievement from a remarkable man. Cannabis became widely available, and the standard British pharmacopoeia product and in America was this thing called tincture of cannabis. It's simply an alcoholic extract of the female flowering heads, or the resin. And you can see it's made by the company that Ray and I used to work for, or rather Sharp and Dome part of it. We used to work for Merck Sharp and Dome, but this was the original Sharp Dome company in America. And tincture of cannabis was widely available over the counter and claimed to treat lots of different diseases and present in lots of proprietary medicines. For example, you could go to a Victorian pharmacy and buy all sorts of things over the counter that you couldn't dream of buying today. You could buy morphine, opium in various different preparations. You could buy cannabis in all sorts of different preparations. And one that was particularly popular was Dr. Collis Brown's chlorodyne. This was an absolute bestseller in Victorian England and went on for some time into the 20th century. And it's no wonder that it had such wide indications: coughs, colds, you can read some of it here, cholera, dysentery, diarrhea, all legitimate uses for this compound, because it contained about 50% tincture of cannabis and 50% laudanum, which is an alcoholic extract of opium. And together with that, a certain amount of chloroform. So the claims made for it were actually probably quite accurate, that it was a cure-all for almost all diseases at a time when you have to remember that modern medicines just weren't available. How many people became addicted to chlorodyne, or how many suffered, we'll never know, because addiction wasn't even thought about in those days until late in the 19th century. But eventually it dawned on some smart doctors in the BMJ article here. In 1974, it took a long time to dawn on people that the morphine contained in the Collis chlorodyne mixture was probably quite dangerously addictive. By that time, the cannabis had been more or less removed, but there was still a good dose of morphine to be had. And these doctors cleverly observed what they thought were 8 cases of chlorodyne dependence in London drug clinic, and they described the symptoms as being just like those seen in classical opiate dependence, and estimated that as many as 1,000 young men in London might be addicted at the time. But it hadn't occurred to anyone that selling a medicine, this was still not a prescription medicine even, selling an over-the-counter medicine that was sufficiently potent to lead to opiate dependence and tolerance was not a very good idea. However, you can go to your chemist tomorrow and you can still buy Collis Brown's mixture. It's a poor shadow of its former self, but it does contain a small amount of morphine, and it does treat diarrhea quite effectively. And in addition to the small amount of morphine, which is hardly worth having for a heroin addict, there is a large dose of peppermint oil, which is also quite good for the digestive system and would make it quite painful, I suspect, to try to inject this stuff intravenously. So, but they are. A good product never dies. It always finds a new life somehow. But let's get back to the science of this. What is it about THC that makes it so interesting and potentially useful medically? Well, THC, like a number of other plant products, including morphine, although they are plant chemicals, they're recognized in the mammalian brain, in the human brain, by a specific set of receptors, that is, proteins on the surface of nerve cells and other cells in the body. And there are, for THC cannabis, there are 2 sorts. There's the CB1, which is principally the type you find in the brain, and thought to be largely responsible, if not entirely responsible, for the intoxicant effects. And then there's another one called the CB2 receptor, which is found mainly in peripheral tissues in the immune system, on different cells in the immune system, and may have THC-like chemicals, which I'll come to later, may have a role as immune system messengers, in addition to the role of this system in the brain. Now, we happen to have, for research purposes, a very useful compound developed by the French company Sanofi, a compound called rimonabant. I'll have more to say about that later. But rimonabant turns out to be a very selective CB1 receptor antagonist. And to a pharmacologist that love to have antagonists, they can try and find out, by using the antagonist, what functions the receptor may have normally. And we can say the rimonabant blocks essentially all the psychoactive actions of cannabis. In experienced cannabis smokers, given rimonabant, they hardly feel a thing when they smoke the cannabis cigarette. And also the pain-relieving effects of THC are largely blocked by rimonabant. And you can also manipulate the system in other ways nowadays by generating strains of mice that have no ability to express the CB1 receptor, so they grow up without any CB1 receptors. And those mice no longer respond to the pain-relieving effects of THC. And presumably they can't get high at all. THC itself has been around as a prescription medicine, surprisingly, for quite a long time. It's got two indications in the USA. One is treating the wasting syndrome in AIDS patients who tend to lose appetite and literally waste away. And among other things, THC is an appetite stimulant. It's also used as an antiemetic. It's got powerful anti-nausea, anti-vomiting properties. The synthetic compound, you see at the bottom here, nabilone, is one of the many synthetic chemicals made by pharmaceutical companies. After THC structure was discovered, the pharmaceutical industry tried to make a medical version of THC which lacked the intoxicant properties. And there are hundreds of publications in the Journal of MedChem and other chemistry journals about describing a number of very potent cannabis-like, not cannabis-like chemically, but cannabis-like pharmacologically, acting on the same CB1 receptor. And this is just one of them. It happens to have some medical use and is approved also as an antiemetic, although very, very little used. Neither of these compounds have been very much used, although they're available as prescription medicines, have been for 20 years or more. And the reason they're not used is taking THC by mouth, orally, as these preparations are. The preparations are THC dissolved in sesame oil or some other vehicle in a gelatin capsule, and you swallow it. And then the absorption after oral administration is very unpredictable. Depends on whether you've just had a meal, whether it was a fatty meal. So you can get different absorptions in the same person from one event to another. And it's generally very unreliable. It's very slow. Takes several hours to reach peak plasma concentration. And generally, patients find it's better to smoke herbal cannabis because it's much quicker. However, they've been there, and they are there. And the revival of interest in this area is described in a very good document that, produced by the Wellcome History of Medicine just last year. If you're interested in the recent history of how cannabis as a medicine suddenly came back into favor, particularly in this country, you should try and take a read of this. It's very, very good. And it has been quite a remarkable story, because we are now, partly because the government, to everyone's surprise perhaps, was particularly lenient, and maybe because of the House of Lords inquiry in 1998 that Walter Perry chaired, and I was the scientific advisor at the time, and produced a report saying that there were probably medical benefits to be had in cannabis. And this gentleman here, Jeffrey Guy, shown with his cannabis plants in the legal greenhouse somewhere in the south of England, that's him with his female cloned cannabis plants. And the powers that be in the Home Office and the various other government departments involved were very receptive to the idea and gave him permission not only to set up a growing facility, but also gave him permission to do clinical trials, which is quite a big step forward, because this is, remember, a Schedule 1 compound. Schedule 1 means this compound is dangerously psychoactive and has no medical indications. So for a Schedule 1 drug to be permitted to go into clinical trial, that was a big step forward. As part of my research on the subject for this lecture, I went to the growing facility when the plants were a little bit higher. I wasn't high at the time, but there are the plants very, very beautifully cultivated, huge amounts of cannabis resin in the female buds. And this is a very carefully controlled scientific process. The plants are cultivated under optimum light heat conditions. They all come from the same genetic strain, as I've already said. They're all female. And the yield of cannabis can be controlled very precisely. I think in this case, around 10% THC plus or minus 0.2% standard error. So this is scientific stuff, and the extraction doesn't involve dangerous chemicals and so on. So it's an industrial process. And by the way, I don't have any shares in GW Pharmaceuticals, the company doing this, nor am I a consultant to Jeffrey Guy. I simply went there for interest to see what it was like. One question about medical cannabis is how can you deliver this very insoluble material? We're talking about THC, really. And smoking turns out to be a very efficient way of delivering a drug of this type, as it is indeed for other relatively insoluble compounds such as heroin. If you heat these compounds, heat cannabis in a cigarette, at about 200 degrees, it volatilizes, the chemical turns into a vapor, and then it turns into droplets, minute droplets as a smoke, and the smoke can be inhaled into the lungs by the smoker, and the lungs, as you know, have an enormous surface area absorption field, and this delivers the drug very quickly. As you can see here, this is how rapidly the plasma concentration goes up after beginning smoking, and you can repeat that a second time and get much the same performance a second time around, and you can see it goes up quickly and it lasts a reasonably long time. So this is very effective, and that's why in medical uses of marijuana or cannabis, most patients prefer to smoke, as they do in America, and as I'll show you later. But there are drawbacks to this. The herbal cannabis smoke contains a whole mixture of chemicals other than THC, of course, and the smoke composition is very similar to that of a tobacco cigarette smoke, and indeed even contains some of the same carcinogens that we know are dangerous in tobacco smoke. So far, there's been no evidence to link cannabis smoking with lung cancer, but who knows? It took almost 50 years to work out that relationship between cigarette smoking and lung cancer, and cannabis hasn't been around that long. But so far, no real problems of that sort. Nevertheless, cannabis smokers do develop irritating coughs, they have bronchitis, they tend to have irritated airways, and these are things that might be avoided if you could find another way of delivering the drug. One idea that's been tested and looks as if it might be possible is to use a vaporizer, that is an instrument that doesn't burn the herbal cannabis but simply heats it up, produces a vapor and a smoke that can be inhaled. But the problem with that is you have to heat it above 200 degrees to get volatile THC, and lots of other plant chemicals will come into that smoke as well. However, that may be possible. GW Pharmaceuticals have used another way of doing this by making an extract of the herbal cannabis in a solvent, and then using this type of metered dose spray under the tongue into the oral cavity. There's quite rapid absorption. You're not swallowing, so it's not going through the lengthy process of absorption in the gut. It's more rapid. It's not quite as rapid as smoking, but it's a pretty good compromise. Well, if you think about it, if you want to introduce medical cannabis in our hospitals, for example, our hospitals are all now no smoking areas. So you're going to have a smoking room for cannabis use? I can't see that. So this would be a more acceptable way and a more precise way of delivering an exact dose. Each time you push the button here, you get 100 microliters and a fixed dose of THC or THC plus cannabidiol, which I haven't mentioned so far, but cannabidiol is one of the relatively inactive, well, it is inactive on the CB1 receptor, but it's a major component of the cannabis extract. There's about as much of that in the extract as there is THC. And some people, including Jeffrey Guy, GW Pharma, believe that it has a modulating effect on the THC pharmacology. But that's what it does, and that's what happens in the clinical trials of this product. Patients are given this type of device, and they are asked to self-administer And the interesting thing is the patients find a level of how many puffs they want to have every day, and they avoid intoxicant levels. Most patients with diseases such as MS don't want or are actually frightened by the intoxicant effects, and they will avoid them. So each patient tends to titrate to a certain level, and remarkably enough, they stay pretty well stable at that level for days or weeks on end. We are now in possession for the first time of some proper clinical trial data. You start off with a group of patients you're going to treat. You randomly allocate them to either a treatment group or a control group who receives the same looking tablet or capsule but doesn't contain any active drug. And these got to be randomly allocated so the physician can't bias the sampling by looking at for patients that look likely to respond as opposed to those not likely to respond. Then they go into the period of treatment. And the treatment has to be done blind, double blind. What that means is neither the patient nor the doctor or the nurse administering the medicine knows whether what's being given is the active material or the placebo, the inactive. And that's important too, because if the doctor or nurse knew they were giving patients active drug as opposed to inactive, they might encourage the patients by various body language signs or whatever to think they're getting better. So that's the way we do it nowadays. It's the gold standard, and at the end of the day, we ask the question, what was the outcome? Yes or no, or somewhere in between. And I'll show you in a moment what happens when we do this, when preparation like Sativex is used in this way for example, controlling pain. Pain relief is one of the real interesting indications for THC or cannabis. It works in various animal models, especially in animal models where peripheral nerves are damaged, neuropathic pain due to nerve damage. And those effects are all blocked by rimonabant or in CB1 knockout mice, as I've already said. And there are some interesting interactions with opioid systems, which also, of course, can yield powerful pain-relieving effects. And there appear to be some, although these are distinct mechanisms, there appear to be some synergism so that a small dose of THC will help produce a larger response to a given dose of morphine and vice versa. So what happens in a clinical trial? Well, here are some typical results for Sativex, the herbal extract given to patients suffering from central neuropathic pain in multiple sclerosis, a very nasty condition. Multiple sclerosis, as you probably know, involves damage to the insulating layers of nerve tracts, rendering them dysfunctional. And in the peripheral nerves, this can lead to limb spasticity, stiffness, and can lead to very difficult to control pain. And when this happens in the brain, in tracts in the brain, there's a central version of this nerve damage pain which is even more difficult to treat. Doesn't respond to morphine, for example. But here's a typical clinical trial using the double-blind placebo control. And isn't it interesting, the placebo group, shown at the top here, actually show quite a significant improvement. And this is quite typical of pain trials, quite typical of CNS trials. Patients who receive the placebo, not knowing whether it was active or inactive, are somehow telling their brain systems to generate opioid peptides and get a certain degree of pain relief. At least that's what we think is going on. If we could only bottle up the neuropathic pain placebo and sell it, it would be a bestseller, but maybe that's called homeopathic medicine. The positive result is the group treated blind with Sativex showed a somewhat bigger pain relief. How do you measure pain? The only way you can measure pain is to ask the patient, how are you feeling today? How is your pain? You usually show them a ruler with a 10 or 11-point scale, 11 point because it goes from 0 to 10. And try and put your pencil on where your pain is at today. Is it no pain at all, 0, or is it the worst you've ever had, which would be 10? And these patients start out around 7, and the pain goes down a little bit. And the difference, the key for the clinical investigator is, can you show a difference between the placebo dummy and the active drug? And in this case, the answer was yes, although it's a very modest difference. about 1.25 points on an 11-point scale. You might say that's not much. But patients subjectively say that's clinically meaningful. And the regulatory agencies will just about approve that level of pain relief, although it's sort of borderline. Not so borderline, though, that in Canada, this type of study was enough to get approval for Sativex a few years ago for treating pain associated with MS, multiple sclerosis. And this has been on the market in Canada now for some years as a prescription medicine. So this is the type of stuff you have to do. You have to do preferably large numbers of patients to get meaningful results. And that's exactly what happened in multiple sclerosis. The MS is one of the real important indications where there are a number of unmet medical needs not treated by modern medicines. And there are a number of animal models that try to mimic the demyelination associated with human MS. And the animal models do exhibit muscle spasm and rigidity and pain. And in addition, we have a lot of anecdotal evidence for patients suffering from MS who actually illegally smoke herbal cannabis. Some of them grow it in their backyard. And the MS Society did a survey of their members some years ago and found something like 5% were doing that and claiming real benefits. One particular lobby group lobbied very strongly for medical development of cannabis on the basis of that type of self-reported use, anecdotal. But we know as scientists that anecdotal evidence doesn't impress any government regulatory agency. So you have to do proper trials of the sort I've described. And fortunately, in this area, the Medical Research Council sponsored the first really large-scale trial of cannabis and/or THC in MS patients with a total number of 630 patients. This is by far the largest clinical trial ever done anywhere in the world for cannabis or cannabis-related medicine. And I'll show you some of the results. The results were a bit patchy. There were 3 groups of roughly 200 patients in each group, a placebo group, one group receiving herbal cannabis extract, and one group receiving pure THC, and all dressed up in the identical-looking capsules, so you couldn't tell which was which. And what happened was that the patients reported subjective benefits. Maybe I should show you the actual results here. Reported some benefits at the end of 6 months, subjective benefits both in terms of reduced pain, reduced spasticity of the limbs, and improved sleep, which is also quite important for MS sufferers. But the objective measure done by the clinicians on a so-called Ashworth scale was manipulating the limbs and trying to measure the degree of stiffness on a 4-point scale, the so-called Ashworth scale. That is rather a blunt instrument, and it turned out to be very ineffective. You get a wide range of results. The horizontal line represents the placebo, and the plus and minus over the placebos is all over the place. A large variation in results among patients, even though there are 200 in each group. And there was no effect statistically at 6 months. But lo and behold, at 12 months out here, there was at least one group showing a statistically significant benefit, objectively measured, not asking the patient to self-report. And that, strangely enough, was with the pure THC group, not with the herbal cannabis extract. Those people that believe in herbal medicine were a bit disappointed by this, but pharmacologists may be quite pleased that the answer is so simple. So that really does support the use of THC, at least, in MS, spasticity, and the Canadian data show also use in treating pain associated with MS. But there are a lot of other actions that THC has. It's a powerful antiemetic in both animals and people, and was used quite widely as a prescription medicine available for treating nausea and vomiting, for example, associated with cancer therapy, chemotherapy. Cancer chemotherapy is dreadfully associated with nausea and vomiting, very severe, may last for several days after the chemotherapy ends, and it's very distressing to the sufferers. And THC is one of the ways in which this can be treated quite well. But again, patients tend to use smoked marijuana or cannabis rather than the oral preparations. And nowadays we have lots of other medicines available which are very effective in treating this nausea and vomiting. Pleased to say one of those is a substance P antagonist, at last found a use for this neuropeptide. And Ray was instrumental in developing that drug and having it on the market for Merck as the first neuropeptide substance P antagonist which had a real medical use. But of course we could use 5-HT3 antagonists as well, and nowadays these are often used in conjunction. So we don't really need THC anymore with all the complications about delivery and intoxicant properties. So this is rather little used. Then there's the stimulation of appetite, and there's still a use for that in AIDS patients, although again they prefer to smoke rather than take the oral tablet. And there are, I'll show you, sorry, getting ahead of myself a bit here. There are other actions which we can see for possible actions for cannabis antagonists, such as rimonabant. Rimonabant was developed by Sanofi Aventis, a large pharmaceutical company, and they've found, not surprisingly you might say, because we've said already THC stimulates appetite, not surprisingly rimonabant inhibits appetite. But in addition, it has actions at a variety of sites outside the brain. The appetite regulation center is thought to be a hypothalamus center in the brain. But outside the brain, in the fat cells themselves and in the liver, there are also CB1 receptors, and the overall effect of rimonabant is thought to be due to combination of central action and peripheral action. And this is the compound for those people that like chemical structures. This is rimonabant. And here are some of the data on weight reduction. The indication here was to treat people with morbid obesity and try and prevent the development of the so-called metabolic syndrome, which these patients tend to develop as a prelude to developing full-blown diabetes. So that was the indication. Sanofi actually called it treatment for the metabolic syndrome. They didn't mention obesity, didn't mention appetite. These are dirty words in the pharmaceutical industry because there have been so many failed attempts to treat obesity with appetite suppressants. But anyway, here are some of the results. And they look pretty good. This is a placebo at the top, 5 milligrams once a day, 20 milligrams once a day, every day for a year. And these are tested sporadically during the year. Large cohort of patients. The patients are maintained on a restricted calorie diet, all of them. And that has some effect on the placebo group. But you'll see you get quite a large effect with 20 milligrams of rimonabant. You're losing at the end of the year something of the order of 6 kilograms. The reduction in fat comes largely from the abdominal visceral fat, which is thought to be one of the high risk factors for heart disease. And you're losing here about as much as 6 or 8 centimeters, which is quite nice to have also for many people. So this all looked wonderful. On the downside, however, on the right-hand side, if you go on doing this for another year, as long as you keep taking the drug, you're okay, you're down here. But if you stop taking the drug, your waistline creeps up to exactly where it was before. Your body weight creeps up to exactly where it was before. And I'm afraid this is typical of all pharmaceutical treatments for obesity and weight gain. Same was true for the amphetamines in the old days. Take the amphetamine, you lose weight, you lose appetite, you lose weight, and then you stop taking it and you go back up again to what seems to be something like a set point, which is hard to avoid. But that was the data. And Sanofi Aventis accumulated data on tens of thousands of patients, both sides of the Atlantic. Other pharmaceutical companies leapt into this, seeing dollar signs before their eyes for the first safe treatment for obesity and metabolic syndrome. Merck did a big program, Pfizer did a big program, most of the major companies. Quite easy to find a CB1 antagonist of their own and start clinical trials. And they all did that, but it all came to nothing because a small proportion of patients treated with rimonabant developed adverse psychiatric reactions, anxiety, low-level psychosis, and worst of all, suicidal thoughts. And in Europe, the drug was on the market briefly and then withdrawn in Europe about a year ago, or February '09, because of the possible link to suicide. Never actually proven to be cause and effect link, but sufficiently worrying to cause the European Medicines Agency to bring this off the market right away. The US had already been cautious. The FDA had not approved the drug in America because of these psychiatric side effects. And all the major companies that were doing clinical trials encountered something similar or gave up anyway because they saw they were fighting a lost cause. And by that time, several $100 million have been spent. Pharmaceutical development doesn't always run smoothly. Let's take a look at snapshots of medical cannabis around the world. Well, the Netherlands, as you know, had a very liberal policy towards cannabis, both medical and recreational. And in Netherlands today, you can get prescription for herbal cannabis. Pretty good stuff, has about 18% THC content, cultivated herbal cannabis. The indication for herbal cannabis is, again, MS and certain other types of pain and certain other neurological conditions. I should add that in Canada, Sativex is approved not only for MS-associated pain, but also for cancer-associated pain. So it has two indications. In Canada, you can get a prescription for smoked cannabis available, although apparently it's not very good quality and people don't like it very much, and Sativex is approved for these two indications. Now, the situation in the USA is very, very complex. You know, the USA, in general, has had a very hard-line policy on cannabis, and the drug czar has been known to say this should never be approved as a medicine because it gives the wrong message to young people that it's safe. Nevertheless, voters have voted differently, and voters in various states where they can add things onto the ballot, propositions onto the ballot, have approved cannabis pharmacies, which provide cannabis, herbal cannabis, to patients as long as they come along to the pharmacy with a doctor's prescription for whatever indication the doctor thinks they might need it for. And apparently in California, where this started, there are now more cannabis pharmacies than there are Starbucks coffee shops. So it's true. It's an extremely popular new industry. But there are problems. The problems are there's no legal source of supply, and the quality of the product that you get from different pharmacies is not controlled in any sort of way. So that the smoke material you get might have a low content or a high content of THC, and that's very, very uncontrolled. And the federal US government still maintains this is a totally illegal substance. It shouldn't be used at all. It's a Schedule I substance still. And what are you doing providing it with prescriptions in cannabis pharmacies? So that several states are having a battle with the federal government, and occasionally the federal government will send out raiding parties, as they used to do for the Indians in the old days, and break up the pharmacies. But President Obama said they shouldn't do that, so that doesn't happen quite as much. Nevertheless, there's still a lot of reservation about this whole thing. And for example, in Oakland, California, the city government voted recently to license a growing facility to provide good quality cannabis to the pharmacies. And at the same time, the state would make a tax revenue on it. So since California state and cities are desperately short of money, this seemed like quite an attractive idea, but it was shot down by the federal government again, who said that if anybody did that, they'd come out and bust up the growing facility. So this is still a standoff. Nevertheless, an enterprising company, Cannabis Science Inc., is set up, which has the mission of trying to provide more standard medical-grade herbal cannabis. And the burden of public opinion seems to be swinging very firmly in favor of allowing cannabis medical use of pharmacies. And this is widespread. 15 states already approved, and other states are considering ballots on the same issue. So the situation in the US is fluid. California, where this all started, has a huge number of cannabis pharmacy and users. And the state government decided to try and do something about research to provide some real data on the benefits or otherwise of smoked cannabis, because you know, as with MS or any other indication, there's very little scientific data out there. There's a lot of anecdote and a lot of belief, but very, very few clinical studies. So they set up this center and provided a research grant of about $9 million in the year 2000. And that's been dispensed to various investigators around the state. And there have been proper controlled clinical trials, relatively small scale, but properly controlled, of smoked cannabis. Now, it's quite difficult to do a trial of smoked cannabis because any cannabis smoker will tell you that the herbal material has a very characteristic smell, and it's hard to cover that up, and for the patients, it's rather easy to tell the placebo, perhaps, from the active reefer. But nevertheless, it can be done. There are various ways of disguising that with flavoring agents. And the trials have been very useful. There are 5 studies completed so far. They confirm what we already thought, that smoked cannabis is of benefit for pain relief and spasticity. And some of the results in one of the California studies are shown here. This is a rather different design from the one I told you about before. This is a so-called crossover design. You take a group of patients who are first treated with a placebo, and then either treated again with placebo or treated with active drug. These are the placebo groups in each case, and this is the active drug. And you see there's in this case, a significant reduction in the Ashworth scale, limb spasticity, the one that failed to show a result in the British MRC trial. But here's some real data. So that's been useful. And meanwhile, in this country, the situation still is that illegal herbal cannabis is used by many patients, particularly those suffering from MS, who absolutely convinced that it does good for their symptoms. We have the results of the clinical trial that I showed you earlier, which did show efficacy in an objective measure, and in all cases showed subjective benefit in terms of the patient's reports of improved pain and spasticity and sleep. And meanwhile, Sativex, the standardized medical product, has been approved as a prescription medicine by the Medicines Regulatory Agency just now in June 2010 for treatment of spasticity in MS, although it's not yet on the market. Still has to pass the hurdle of the NICE review and whether the NHS can afford it and does it really work properly. Nevertheless, it's approved, and that is really quite a significant milestone, because remember, this is still, at the moment, Schedule 1 material, intoxicant with no medical use. Now we're saying, even the hard-nosed regulatory agency is saying, it does have medical use. It's going to have to be rescheduled for medical use, and it will be on the market. And I suspect there won't be much abuse potential, because as a new prescription medicine, it's going to be quite expensive, and you can get much the same intoxicant effects by buying conventional street cannabis. So I don't think it's going to be a dangerous epidemic of Sativex abuse, but it is an interesting medical advance. And again, please remember, I'm not associated with this company. I'm just telling you the story. Meanwhile, towards the end of my lecture, the reason why we have cannabis receptors in our brain has puzzled a number of scientists. As soon as these receptors were discovered, they're clearly very elaborate pieces of protein machinery that respond to a plant product. Now, you had the same situation with morphine, another plant product, with opioid receptors, several different types of opioid receptors in the brain and elsewhere, recognizing an exotic plant chemical. Now, what's that all about in terms of evolution? We didn't evolve our brains surely to recognize plant chemicals. And sure enough, we didn't. We have our own naturally occurring opioids in the form of opioid neuropeptides, enkephalins, endorphins, etc. And as you see here, we have a whole family of lipids mediators which recognize the cannabis receptors in our brain and elsewhere. Some of these are both CB1 and CB2 active agonists. And there's not just one. The first one to be discovered was anandamide, which was again discovered by the Israeli group in large-scale extracts of animal brain. They isolated an active material that was active on the CB1 receptor binding site in brain homogenates. And Roger Pertwee in Aberdeen was the first pharmacologist to show that anandamide also had activity in a variety of the smooth muscle, in vitro smooth muscle preparations, such as mouse vas deferens, well known to classical pharmacologists. It was active biologically also in the same way as THC. So that was a real discovery. Anandamide was the first one, called anandamide after the word for bliss, or in Sanskrit, I think. Anyway, then lo and behold, we've got a whole slew of them. We've got 5 on this slide, and there are 8 different arachidonic acid derivatives, all of which have the same property of acting on CB1, CB2 sites. What a future generation of PhD students and postdoctoral graduates and scientists will show is why on earth do we have so many different ones? What do they all do? Why do we have different ones in different regions of the brain? All that is really a blank canvas at the moment to a large extent. But we can see pharmaceutically some interesting possibilities. If we can invent drugs to manipulate this endogenous, naturally occurring system, they might have some of the beneficial effects of THC or cannabis in a more focused way. For example, we know that these substances are largely degraded by either enzymic processes or tissue uptake processes. You can inhibit the transport process, or you can inhibit the enzyme. Pharmaceutical chemists are pretty good at making enzyme inhibitors, and this is already happening. And some of the inhibitors of the enzyme fatty acid amide hydrolase, FAAH, are already showing interesting pain-relieving effects in animal models. And you can see the parallel here with monoamine uptake inhibitors as antidepressants. By inhibiting the transport mechanism or the enzyme, you would prolong the actions of endocannabinoids only in those regions where the system was being active. These are not chemicals that are stored in nerve endings. They're more like prostaglandins. They're made on demand, and their synthesis is rapid. And since they're small, easily diffusible molecules, they reach their targets very, very quickly and easily. So those parts of the brain or the periphery where this is happening would be targeted selectively by drugs that were to inhibit their inactivation. Conversely, you could think about drugs that inhibit the biosynthetic enzymes. So there are a lot of possibilities there for the future, and many of these are being actively pursued already. And I'm sure we will see a new generation of cannabis-based, endocannabinoid-based medicines coming in the future. But Iverson's rule is that it usually takes 20 to 30 years between a laboratory discovery such as this and a new medicine being available for patients. So I don't know if I'll live that long to see this happening. But it's got tremendous potential, and that's just summarizing what I've already said. There's also the possibility of making metabolically stable analogs that would prolong the life of compounds like anandamide that are otherwise very rapidly inactivated. And there's a certain amount of interest in CB2. The CB2 receptor as an anti-inflammatory potential pain mechanism is being pursued also, and drug companies like the CB2 receptor because there's no danger of having a psychoactive side effect, because this is not associated with the intoxicant properties of cannabis. And finally, if you're very optimistic, you can think about all sorts of human conditions that might be treated by cannabis-based medicines. I'm not saying I believe in all this, but there are a number of indications I haven't had time to talk about. But anyway, I think I've finished, and I'll stop there. Thank you.